Hello students, welcome to lecture 6. In this lecture we will go into more details of event-related potentials, which are one of the prominent components of EEG signals. The outline of today's lecture is as follows. First, we briefly review about evoked potentials, definition of event-related potentials, and oddball paradigm. Also, we will see averaging cross trials helps to extract useful ERPs, event-related potentials. We'll also briefly visit receiver operator characteristics definition. Let's provide some formal definition of evoked or event-related potentials. So ERP, in short, event-related potential, is an electrical brain activity that can be measured by EEG signal. One of its important characteristics is that it's a time-locked event, or to some event. It's a time-locked response or potential to some event. Okay? The event may be external sensory stimulus or internal or associated with the execution of motor, cognitive, and psychological task. So external sensory stimulus include visual, haptic, auditory stimuli. It can also be generated internally or endogenously with the execution of motor function or intent of a motor movement will generate a ERP event, also known as readiness potentials. A subclass of ERPs are called evoked potentials. They reflect the processing of physical stimulus rather than relation to any kind of higher processing in the brain that may involve memory, expectation, or attention. So evoked potentials usually related or reflect the processing of stimulus, physical stimulus. ERP waveform is typically composed of several ERP components. This is example of ERP. P1, N, P2, N2, P3, each of which is related to a positive or negative voltage deflection. And as you can see, the ERPs typically have a label that starts with a letter P or N, which indicates the polarity of the deflection. It is also followed by a number indicating either the latency, 1, 2, for example, in milliseconds, or component's ordinal position in the waveform. P1 comes first, N2 comes first, so first positive, second positive. Okay. And some ERP components are rather labeled by also acronyms that refer to their function. Right? For example, PE sometimes is labeled PE, positive deflection, related to erroneous decisions. NE, for example, we may have some NE label, okay, which reflect negative deflection related to erroneous decision. Can also be noted by ERN, event-related negativity. Well, also we can see that lateralized readiness potential, which reflect the preparation of movements, motor movements, can have similar shape with corresponding labels. We can also see some other extensions or mixed labels like N2PC, which is a component like N2 originating in the posterior scalp location, contralateral to the side of direct attention. Visual evoked potentials are also ERP events, ERPs in EEG, that are caused by stimulation of subject's visual field. Visual field, as you can remember, is in the occipital lobe, visual cortex. So commonly used ones are, as a stimuli, checkerboard stimuli that flip at an inter-stimulus interval of 1 to 3 seconds. So the subject is shown with the flashing checkerboard patterns. And more specifically, these are called pattern reversal visual evoked potentials, in contrast to many variants of VEPs. So visual evoked potentials can be observed regardless of attention, but the amplitude depends on the intensity, size, and focality of the stimulus. So experimental paradigm or parameters of the stimulus. So let's take a look at how these event-related potentials or event-related segments of continuous dataset look like. This is example of one trial or example of event-related potentials acquired at PZ, parietal lobe of the brain, and we have the amplitude value, which range from microvolt, and also time. So 0 usually corresponds to the onset of stimulus. So when the subject sees a visual or some auditory stimuli, and which is followed by this kind of pattern. So how do we acquire these event-related segments from continuous? Okay, for example, here we have continuous EEG signal acquired from one channel, PZ, for example. And during the experiment, okay, the visual stimuli is shown to the subject, and then time related to the onset of the visual stimuli is recorded along with the data. So we can use these kind of time markers when the visual stimuli is shown to segment the data into epochs or segments, okay, from continuous into segment. So we call them also epochs around the stimulus time marker. So for example here, K1, 2, K3, K4, K5, K here denotes the number of observations of the ERPs. And if you take all these epochs and perform averaging, we can see the following pattern. This is the average from these 5 segments. And in BCI, we work with this kind of data set, BMI. So let's try to visualize single trials of event-related potentials. So here we see the single channel CZ recordings of event-related potentials. So with different colors, so different colors are superimposed. And then we can see these average ERP acquired over 150 trials. We can visually say that the data look highly variable. There seems to be a very big inter-trial variability, but the average ERP tries to capture the common deflection or changes in the data across all 150 examples. Let's try to represent ERP using mathematical notations. Okay, because we are dealing with the data and we need to perform some mathematical operations on that data. So in these coming series of lectures, we will denote scalar values as the italics, x, n, or gamma. Okay, this will represent a scalar value, while vector, the column vector, will be bold-faced or lowercase bold-faced, okay? And we represent matrix as bold uppercase, bold-faced uppercase, so X or sigma. This will mean that we're dealing with matrix. So for matrix X, okay, the term Xij denotes elements of X in the ith row and jth column. Ith row and jth column. And for finite set, okay, we denote its cardinality by the following, right? Similar to the absolute value. And we most of the time assume that EEG is a random variable and has some underlying distribution. Oftentimes we assume that the EEG are independent and distributed according to Gaussian distribution or normal distribution. N is a distribution with the following mean and variance, right? And with the following following halts, for example, the alpha scalar X random variable will also be reflected in the distribution. And sum of all separate individual random variables or trials will also be reflected in the distribution of the data. We can now try to represent or provide a model for ERP. So generally the SNR or signal to noise ratio in EEG is very poor or bad. As an investigator, you are interested in few specific signal components that are related to some event or time lock. For example, those P1 or positive deflection or negative deflection. However, the brain contains millions of neurons, right, that produce signals all the time. Okay, we call them background activity. Any activity that's not relevant to the signal of interest or to the experimental condition, we can call them as background activity. And reducing this noise is a big challenge in EEG analysis in general. One common technique in ERP is just acquire a lot of separate trials of examples of ERPs and then perform simple averaging. This can extract the signal components that are related to some event we are interested. Let's consider ERP model. Okay, let's call ERP as P of t, potential at time t. That is superimposed linearly, okay, superimposed by background brain activity. Let's call it as R, as in residual or noise. Then we can represent EEG, acquired EEG, as X of t that consists of the potential or event-related potential plus some noise. This is a model that we typically use in representing the EEG data. Let's now assume that ERP, the signal of interest potential, ERP potential, is constant in each trial across k trials, while residual, the noise component of EEG, is generated IID according to some Gaussian distribution with zero mean and variance of 1, for example. So IID stands for independently identically distributed, okay, for fixed. So what we can observe in this visual example is that averaging actually helps if our assumption is correct or is valid. Well, we record EEG signal, which consists of ERP plus noise across several trials, okay? So we see K1, K2, K3, K4, all representing different ERPs, okay, EEG signal, the segmented EEG signal. And then if you assume P of t is constant and noise is ideally distributed, we can recover we can extract event-related potential by simple averaging, which is as you can see, very similar to this one, right? The assumed P of t potential, and noise also. Here we see some amplitude variation change. If you perform averaging, noise also gets eliminated on average signal. So visually we can say that the averaging helps if you assume that PT is constant and IID. Alright, mathematically, x of t is equal to P of t plus noise across k trials. Then averaging across trials, results in the following, leads to this. So we are just summing all the EEG signal across k and averaging, right, by dividing by k, will give us the following relation. So because P of t is as assumed constant, we can take it out from our signal model, plus the 1 over k is noise, right? So the noise in the average will be minimized. In other words, if you average k trials, so 1 over k, the k will be applied to minimize the variance of the signal, right? While the mean is 0, the variance will be multiplied by 1 over k, which in turn will minimize. So we can show it with a simple calculation. Originally we assumed that noise is generated by some Gaussian distribution, normal distribution, with sigma. And if you perform summing, which is equivalent to the following. We just multiply the k to the sigma, the variance. And while division also will minimize the variance eventually. Right? The amplitude of noise goes down by a factor of square root of k in an average across k trials. Visually we can see. We eventually want to minimize the noise in the data and recover the true signal of interest to ERP component. That's why we talk about noise here. So if you want to build a BCI that uses event-related potentials, there must be at least 2 conditions: that the user can voluntarily attain, and they need to be discriminable in single trials, or at least with just very little averaging. These 2 different experimental conditions can be acquired using so-called Oddball paradigm that is used in experimental neuroscience studies. In this paradigm, the Oddball paradigm or experiment, there are 2 kinds of stimuli. For example, low and high tones, green circles or red circles, 2 different stimuli presented to the user, visual or auditory. And stimuli are presented at regular intervals in a random sequence. One of the 2 stimuli is more frequent than the other, most commonly with the following ratio. So one of the stimuli appears 80% of the time, the other 20%. So frequent stimuli that is shown 80% of the time is called standards, while 20% shown stimuli are called deviants, standard versus deviant. The test person has a task to detect the deviant stimuli while being shown, standard or deviant, in a randomized sequence, and count their occurrences, usually silently. So typical example of ERP acquired from the Oddball experiment looks like this. So the deviant that is shown 20% of the time has the following features with increased amplitude, if we compare to the standard. deviant and standard stimuli, related brain response or event-related response. And again, in most of the time, the 0 here corresponds to the time when the stimuli was shown, and then the brain response. Okay, here this is average example in time series form of visualization, while this one shows topographic image. Okay, data, for example, this point corresponds to CZ location, and the colors are representing the amplitude values of the ERP. Okay, more red means higher amplitudes, 15, 10 microvolt. Blue means more or less negative amplitude. And one prominent ERP component observed in this oddball paradigm is called P300 waveform, which is pronounced after deviant stimuli only. So P300 means P stands for positive wave, positive deflection. 300 means the time when it happens, where it is observed. If we assume this point, the origin starts at 0, 300, around after 300 milliseconds, we will see this prominent response of the brain. And most of the BCI applications use P300 waveform for building a binary classification or binary BCI, which we can use for different applications, different spelling, control of robot, and others. But the thing here is that using oddball paradigm, we can get two different types of the conditions that we can use for training our machine learning algorithm for classifiers. Nonetheless, there are factors influencing the P300 waveform. Okay, so in order to be able to obtain features that represent standard versus deviant, they should be separable strongly in terms of some amplitude features or some frequency features. So what has research shown to us is that the shorter target to target intervals, we refer them TTI. Okay, so target to target intervals means when we present stimuli to user, we will have the sequence, randomized sequence of target, non-target, target, non-target events. So deviant will be the target. Deviant to deviant intervals. We can try to, in between deviant to deviant interval, we will have a standard stimuli as well. But let's talk about this as target to target interval. So shorter TTI reduce P300 amplitude, which is not good. Why? Because discriminating these features will be difficult for a classifier. Another thing that has been observed is that task relevance increases the amplitude of P300. So task relevance of the experiment or task relevance of the stimuli or task difficulty of the experiment can also reduce amplitudes. Okay, too complex, too simple tasks reduce P300 amplitude, or latencies are longer for more complex tasks. Another factor is that the attention related, the allocation of attention. If this resource or allocation is reduced, the P300 amplitude decreases and its latency is prolonged. Or lastly, emotional meaning of the stimuli. Okay, for the users can increase P300 amplitude. So all of these factors needs to be taken into account when we want to build a BCI for using P300 waveforms. And our goal is to obtain P300 with increased amplitude, because amplitude is one of the representative feature of P300 waveforms. How can we use Oddball paradigm to build a BCI speller. Let's take a look at this example. So assume that our stimuli is a beat or musical tone with deviant and standard, two different types of the stimuli. So when you provide these auditory stimuli to the user and then record data from the scope, here shown CZ channel. Okay, so these rectangulars represent standard stimuli, and deviant is by red color. Okay, so most standard is presented 80% of the time, deviant 20%. So we have the 80 to 20 ratio. When we record data that corresponds to these stimuli with time markers, we can create or segment data and first visually try to observe if there is any difference between the brain response related to standard or deviant stimuli. Okay, so here are the segments of the EEG signal, shaded. Usually this shaded area is around 600 milliseconds. We call them as epochs or single trials in EEG field or BCI field. And then these trials are grouped into several classes defined by stimuli or standard. So two classes in this case, one class versus another class. We can visually see that, look, there is increase in the amplitude. This is one example of single trial P300 waveform. Note that this data is acquired from auditory stimuli, right? When you hear different notes, you can still get a different brain responses. Well, this also works in the visual stimuli case. For example, instead of hearing different tones of music, let's assume that we have a monitor placed in front of us, and that monitor is showing us sequence of two different kinds of visual stimuli that contains either circles or a square or this kind of a ramp or shaped stimuli. Similarly, we can classify them as deviant, which appears less frequently, and standard. Why it's called deviant? And why it's called standard? Standard is the stimuli that happens more frequently. It's like more standard stimuli. And there is suddenly we see some different stimuli, right? That deviates from the standard. So we are interested in detecting this deviant. And sometimes this deviant is called target stimuli, while standard is called non-target stimuli. In other words, we can relate these target and non-target to attention that the user is paying. So slight modification to the visual stimuli, we can see here circles, triangles, rectangles, and cross, and octagon. Different shapes, different colors. So instead of presenting just binary or two different types of stimuli, we can make it a little bit more diverse. Then the user decides which one of them target and which one of them is non-target based on his or her attention. So due to the attention task, the two classes of stimuli are called target and non-target. So in this case, we have 5 stimuli present with the same probability. One of them is defined target in an attention task. All right. Now, how can we build BCI speller using the data collected from Oddball paradigm? So as I said, we have two conditions, standard and deviant. If you translate those two conditions into spelling, we can have a character of interest that we want to spell. For example, here, brain, N. Then this will be the deviant, right? And then all other characters being flashed to the user will be standard. So instead of the monitor, these symbols, user will be given a sequence of different characters in form of stimuli, right? And then user can classify, or user can define which one is target, which one is non-target, which one is deviant, which one is standard. Then we can collect the data, segment, and build a BCI speller using those features. So intended letter is target and all others are non-target. The distribution of ERPs are also different. If you analyze target and non-target related ERPs, we have the following statistical values of that corresponds to P300 amplitude, so mean and then variance. You can visually see that they are very discriminable. Usually these features are fed into classifier, and then classifier detects whether the data or segment of EEG corresponds to intended target or non-target. The entire pipeline of the BCI classification can be summarized in these figures. Okay, so first we have a continuous EEG required from experiment. It can be Oddball paradigm. Second is segmentation of continuous EEG into trials of predefined class, target, non-target, or other. We will get, as a result, epochs of raw EEG, and then from raw EEG we need to extract features, useful features, okay, that can be used in the classification. So graphically, from continuous EEG with the time markers, where the marker 1 corresponds to standard or deviant stimuli onset. We construct these kind of features or segments of EEG. From these epochs, we need to do one more step of pre-processing, usually cleaning background irrelevant noise in the data and construct the feature vectors from epoch 1 to 12 feature vector. And finally, we apply the classifier, either binary or multi-class, depending on the type of classification we're solving in the BCI context. Alright, so if you have attended any machine learning class, we know that classifier is a function that maps from feature space to labels. Okay, F, let's represent as F function. To binary classification, we'll have two, yes and no, or target, non-target. Let's take a look at simple classification approach. So in order to implement a BCI speller, which is a binary classification task, we need to distinguish between target and non-target, okay, in EEG data. Target ERP, non-target ERP. So from the Oddball literature, we can conjecture that best discrimination between those cases is granted by P300 component. This is where we can see visually prominent or deflection of biserial component and the difference in the amplitude features. Okay, this comes from previous research as well. And another thing is that the P300 has its spatial focus. Spatial means which electrode best represents the P300. If we record data from the user using 64-channel EEG system, not all of the channels carry or show discriminable P300 component, but it has been shown that usually CZ is the best channel, best place to find P300. So first step would be we construct a feature, we take a feature. Okay, let's focus on the amplitude feature of the P300 at given CZ. How can we construct a feature vector? Alright, if you look at this, we also, because we're talking about just single amplitude features, we can call them as univariate features. So here we see the data collected at CZ electrode, see, with two different colors. So the red corresponds, red trials in the background corresponds to target ERPs, while the other corresponds to non-target. Okay, and then these two lines are the averaged responses or ERPs between target and non-target. So you can visually see this line and amplitude being higher than the non-target. In order to construct a feature vector, we can just pick one time point. Okay, here 220 milliseconds, where we see the peak of the P300. While P300 is expected to occur around 300 after the stimuli, but there is some variation. Okay, so it usually can come a bit earlier or a bit later. But in this case, we see that it happens around 220 milliseconds. We just take this time point and map or construct feature vectors as follows. So these dots represent the amplitude value of each individual trials that corresponds to target, non-target, okay? And this cross represents the average of target and non-target. So what we can do next is we just can feed these to the classifier and classify it target or non-target. Call them as univariate feature based on a single individual trials. So each dot here corresponds to one trial, one training example. Okay. Now, given these univariate kind of features, we can observe here that some of the points, some of the amplitude values collected from ERPs appear to be on the other side. For example, here, look at this blue point, which is supposed to be in this side, right? Or this orange. This brings us the question of how can we measure the discriminability of these univariate or single features. In ideal case, we want to have features, okay, to be located, for example, all blue in this region, all red in this region. So one of the metric used for measure of separability is called point biserial correlation coefficient between two univariate features. So it is based on this formula. It's r squared of given two univariate features with a mean value from the feature 1, and then mu2 correspond to the second feature, second univariate feature belonging to the second class, and n, of course, stands for n1 for one class, n2 to two class, target or non-target. Okay, number of samples of each class. So if we calculate, we can calculate this r squared or this point biserial correlation coefficient or r square value. We can see three different cases. So the r square range between, okay, if r square is equal to -1, then we see that the features, univariate features, are separable, very, very well separated. Similarly, when we have r square +1, also we see that the features are separated, separable univariate. While if we observe r squared to be 0, then we see a lot of features mixing up, so they are not separable, those univariate. So in BCI, one of the important question to ask is, as we collect the data, can we make sure that how separable data is, or can we make sure that the data is good, well separable? We can use this kind of measure to analyze if the amplitude values are separable. Otherwise, we may have to do some pre-processing feature extraction so that the r square is maximized to +1 or minimized to -1. Another commonly used measure of separability is called area under the curve, AUC. Right? It's based on maximizing the AUC. This is the AUC curve, for example, where we have a misdetection rate represented in this axis, while true detection represented in this. Okay, so misdetection of ERPs, true detection of ERPs, so targets, or again the targets. So if you take a look at the classifier outputs, how we can see how the curve changes. So given, all right, this point, blue point, if it is correctly detected, then the curve through detection goes up. Second point, another arrow. While here, if the blue point is detected as orange, then we increase misdetection in this way. And then another curve here, right? So curve. Well, the task here would be to make sure, or to maximize the area under the curve between different features. Okay, we can use them as one measure of separability. Well, we can see that when AUC is 0, we can observe the following. So area under the RUG curve, which also means that perfectly separated distributions. Or random AUC will have the following area, so 50.5. The other case is when AUC is 1, then also we see the area under the curve is maximized and we observe perfect separation. But AUC is 0, we still have perfect separation, we just, the labels are flipped to examples. Well, the classifier output from the previous example are 0.7. Looks like this. So in BCI, AUC metric is used in most BCI research because they represent a robust in performance measure, which is non-biased or unbiased. In the above example, we have shown that collecting a univariate feature from single channel, but in real world or BCI or ERP analysis, we use data from multiple channels, and then we should not use or rely on one channel data. Okay, so otherwise interpretation of these ERP components will be hard or impossible. Well, in the coming lecture, we'll see how we can extend those univariate features into multivariate features to represent data from multiple channels, okay, that can improve classification performance much better. For example, in the multi-channel case, we're not only recording a single channel, but from multiple channels. Okay, and then constructing feature vectors from each individual channel as one tall matrix. That could correspond to target event, while others non-target event. Okay, target event. And construct the features. Well, if we visualize those multi-channel features, collected features at some fixed time point as topographic image across different trials, we can see this color-coded distribution of ERPs. So, while in this plot we see data from a single channel at CZ at 220 millisecond time point. And then if you map those amplitude values at the following color-coded or heat map distribution, okay, on the surface of the scalp where each cross corresponds to channel, we can see, okay, different distribution. Trial 1, 2, 3, 4, 5, 6. While trial 4 represents some interesting localization or observation of some more higher amplitude ERPs around CZ. This kind of visualization gives us also information about spatial feature. For example, which channel contains more discriminative or more strong ERPs, okay, at some fixed time point or at some across. We can also collect the average of between 200 and 220 and then try to visualize as follows. So let's take a look at some of the analysis of ERPs from the EEG that was actually recorded from a BCI experiment or BCI spelling experiment. And then let's visualize the data just to have some idea of how separable, how prominent, how visible they are. The first example here is that we can plot these ERPs in a head plot and visualize in the following form. What we have here is channels, okay, corresponding to AF3, AF4, CZ, CPZ, and then this is the head or shape, which is represented by circle, right ear, left ear, nose. So if we plot, visualize the ERPs versus, in particular, target versus non-targets, we can see that the target as shaded in blue wave is strongly different from the non-target waveform, right? And the shaded areas corresponds to the difference between two classes, two different types of brain response. In this kind of plot, we can visually pinpoint, okay, and ask where target non-target is separable. CZ looks like more separable, CPZ, PZ, while OZ, PO7, PO8 has very less separable. Okay, so acquiring data from multiple channels is important, as they also provide some additional spatial features. So spatial feature related to the group of channels that make them discriminable to classes. Another way to visualize and interpret the EEG data, okay, segment that acquired from Oddball experiment, while where we are analyzing ERP, target non-target is as follows. These are, let's call them as heat maps. Okay, heat maps representing the amplitude value of the EEG time series, EEG data. We have here time information marked from minus 200 millisecond to plus 800. So minus means before stimuli onset. If 0 is where the stimuli has been shown, okay, the post stimuli is represented by positive time indicators, while pre-stimuli negative. So, and we see some channel information. Okay, 5, 10, okay, 15 channels here. And we can represent them as color-coded amplitude values. So we see something visually. Some red increased values around 300 and 400 milliseconds. That correspond to target. If we also visualize non-target in this form, usually we don't see this kind of pattern, increase in amplitude. Or we can also acquire multiple trials of these, okay, data that corresponds to target or non-target class, and then calculate the AUC across different trials and see that those P300 waveforms can be discriminably detected. So what we see here is AUC, area under the curve, metric, separability metric. Another way to visualize ERPs are usually done in a grid form. Okay, so here all those 7, okay, 14, 21, 25 channels, right? So target, non-target, we see some shaded as a difference between amplitude values, right? And also we have these color-coded rectangular values that represent the separability measure. One of the examples is R square that we have discussed, R square metric, point-wise, yeah, by zero correlation. So when we have less separability, okay, these are color codes are these colors are less visible. Using this kind of measure separability, we can also tell where the separability is happening or visible, where in time. So another way to visualize ERP target non-target distribution is through topographic images. What is shown here is that these are called topographic images, as I mentioned earlier. That comes from target and non-target, right? How do we acquire them? How do we get them plotted in this form? So first, we can take a look at the time series, okay? Dataset, or time series representation, and just segment or just select the time segment of interest. For example, from 200 to 260. Perform averaging, and averaging results in one amplitude value, right? And then interpolate on top of this surface, which will correspond to the average amplitude at that time point for both target and non-target event. What we see here is they are different, right? Target has some this kind of distribution, while non-target. Second, okay, another time segment of interest. Take an average and plot it as follows through interpolation. Third one, okay, and then fourth one. We can also generate fifth or sixth, right? So these 4 different topography correspond to one time segment average, second, third, and fourth. What we can see here. So one thing to note is that non-target has some very similar distribution, spatial distribution, spatial temporal distribution. It doesn't look like things are happening here. While target, we see here around 400 millisecond, we see strong ERP or P300 waveform, okay, represented as red color, which is very different from the non-target. So we usually use these 2 features as input to classifier, right, for building binary classifier, binary BCI. Also here. So this kind of visualization can help us to focus on the part of the data that contain discriminable feature in the example of classification, if we are interested in classification or increasing the classification of BCI. Well, we can also visualize this topography using different features or different representation of the ERP target non-target. For example, we can calculate the AUC, area under the target and non-target first, okay, non-target ERP, and then interpolate. First segment, second, third, and then fourth. So AUC shows a difference between target and non-target events. Well, here, okay, this is another way to visualize. So to visualize discriminable feature using the AUC matrix. What we see here is that around 300 and 500 milliseconds, we see strong difference between 2 target and non-target related ERPs. Okay. Knowing this kind of features will increase the classification performance of our BCI system. We also call this kind of time and spatial distribution as spatial temporal evolution or distribution of the ERP. So this is the end of today's lecture. We will have more sessions dedicated for hands-on experiments or hands-on sessions where we will be doing EEG ERP analysis and visualization, feature extraction, some other relevant tasks. So today we reviewed ERP Evoked potentials. We discussed about Oddball paradigm and said that averaging across different trials of time-locked event helps, is useful. And also briefly mentioned about receiver operating characteristics. So these are the references for additional resources you can go and consult.